Okay, ladies and gentlemen, we are on to this course ANS 506 Techniques in Animal Reproduction. We are now going to handle Module 1, Sex Determination, Manipulation, and Heat Detection. By way of introduction of this module, you have two units. The first unit covers sex determination, while the second unit has to do with heat detection methods in farm animals. The objective of the module is to enable you to do two things: understand sex determination and manipulation in farm animals, and get to know heat detection methods in farm animals. By way of introducing the first unit, Unit 1, Sex Determination and Manipulation, this unit has to do with how to determine sex of the offspring and then how to manipulate reproduction in birds, reptiles, and indeed the livestock. The objectives include, which by the end of this unit you should know, you should be able to explain clearly sex determination in mammals and most insects, list methods of sex predetermination, and explain the concept of sex reversal. Now, sex. What is sex? Sex is defined as the sum total of those differences in structure and function on the basis of which an organism is classified as male or female. Sex determination. Two main theories have been advanced in explaining the mechanism of sex determination. The first, chromosomal theory of sex determination. Under this theory, sex is said to be determined at fertilization by the sex chromosomes. Each gamete contains the haploid number of chromosomes. In mammals and in most insects, all gametes produced by the female are similar, having X chromosomes. Males, on the other hand, produce two types of gametes in approximately equal numbers. One type bearing the X chromosome and the other type bearing the Y chromosome. In mammals and most insects, the female is known as the homogametic XX sex, while the male is heterogametic XY. At fertilization, a zygote with XX results in a female, while that with XY results in a male. In birds, the female is the heterogametic sex, while the male is homogametic. These are illustrated in the table below. Be careful to see the table carefully in detail. Aberrations of genetic sex do occur either as a result of non-disjunction of sex chromosomes, translocation, deletion, or mutation. In maternal non-disjunction, fertilization of the ovum will lead to formation of an XXX or an XXY zygote. In paternal non-disjunction, zygotes of XXY or XO genotypes result. Such cases of aneuploidy often result in gonadal or endocrine defects. In man, the Klinefelter syndrome XXY is characterized by gonadal hypofunction. In women, Turner syndrome XO is characterized by gonadal agenesis or aplasia. Now, genic balances theory of sex determination. This theory proposes that sex is a phenotypic trait determined by interaction between genotype and environment. Individuals vary in their degree of maleness or femaleness. C.B. Briggs in 1922 proposed the genic balance theory to explain apparent quantitative variability in sexual character. According to this theory, sex is determined by the autosomes as well as by the X chromosomes, the ratio of autosomes to X being the significant relation. In Drosophila, the X chromosome carries more genes for maleness, table 2, which sex actually develops is determined by the balance between the two sets of genes, as you can see from table 2. Other theories of sex determination include genetic dosage compensation, X inactivation model of sex determination. The ZFY gene, thought to constitute the primary sex determining signal, was identified within a very small segment of the Y chromosome by Page and co-workers in 1987. However, the presence of a similar gene, ZFX, on the X chromosome prompted the propounding of the dosage compensation X inactivation theory of sex determination. According to this theory, both ZFX and ZFY produce functionally interchangeable proteins. It therefore means that XY cells would have two active copies of the gene XFX and ZFY, while XX cells would only have one active ZFX copy due to X inactivation. Embryos with two copies would thus develop into males, while those with a single copy of the gene would develop into females. In this way, the theory holds or held that gene dosage determines sex. Though elegant, the theory became untenable when it later became evident that the ZFX gene escapes X inactivation, thus contradicting the dosage compensation model. Sex determination in birds. In order to distinguish animals with female heterogamety from those with male heterogamety, the distinguishing chromosome is known as W in the former, in mammals, Y, while the common chromosome is called Z, X in mammals. Temperature-dependent sex determination in alligators. Many reptiles, including alligators and crocodiles, exhibit no sex chromosome dimorphism. Rather, the sex of the offspring is determined by the temperature at which the eggs are incubated. In alligators, Alligator mississippiensis, incubation of eggs from, as you can see, 29 to 31.5 degrees centigrade results in 100 female springs. As you can see on the table, others result in 100% male offspring from 32.5 to 33 degrees centigrade. And those resulting in different sex ratios, 100 females, and higher mortality rates are as depicted. Sex determination in fishes. Most fishes function either as male or female throughout life, i.e. most are bisexual, or gonochrostic, as opposed to hermaphroditic, a condition in which an individual produces both eggs and sperm at some stage of its development. Establishment of sex depends on sex chromosomes designated X and Y in most species. Usually XX individuals are female and XY individuals are male. There are exceptions, e.g. among the Posilidia, in which the homogametic individuals is male, chromosome designated W and Z are recognized in some of these species. Although the sexes have similar appearances in many species, sexual dimorphism or dichromatism is common in fishes and may be especially well marked in those species with internal fertilization or elaborate reproductive behavior. Occasional hermaphrodites are found in many gonochoristic species as an abnormality, but numerous species are normally hermaphroditic, with some even capable of self-fertilization. Synchronous or simultaneous hermaphrodites have ripe ovaries and testes at the same time, but usually spawn with one or more other individuals, alternately taking the role of male and female. Synchronous hermaphrodites are known from the following families: Chlorothermite, Betheteridae, Alypsoridae, etc. Protandrous condition is known in members of Gonastronidae. Serenidae, Sparidae, etc. Protogynous hermaphrodites have been noted in the Synbrachidae, Serenidae, Menidae, and Labridae. Sex reversal and hermaphroditism are controlled by the endocrine system, which is genetically programmed in normally hermaphrodite species to act on the gonads in response to the proper stimuli, which can be internal or external. Predetermination of sex. X and Y chromosomes bearing sperm have differences in quality of DNA. The X having more DNA than Y chromosome bearing sperm. Bull, boar, and ram, X and Y chromosomes bearing sperm population can be separated by flow cytometry procedures. Measurements of DNA in separated X and Y bearing sperm populations can be used to predetermine the sex of offspring. Centrifuging of semen results in the Y chromosomes bearing sperms being in the upper half of the supernatant, while the X chromosome bearing sperm are in the remaining half supernatant below. In humans, semen deposition into the female reproductive tract at peak body temperature or the sudden fall in temperature in normal female cycle results in a male zygote. Ovulation is said to take place at either peak body temperature or the sudden fall just after the peak. Y chromosome bearing sperm are said to be lighter than X and therefore swim and reach the egg before the Y, but the Y are less viable than the X. Sex reversal, gamete and embryo sexing. Sex reversal is common in some species of fishes as earlier discussed, C 2.4.4. With the development of the embryo transfer technology, interest in the sexing of gametes before AI or embryos before transplantation have become substantial. Gamete sexing differentiating the Y chromosome bearing sperm has been earlier discussed. Possible cytological approaches in sexing of embryo before transplantation are A. Biopsy of bovine embryos on day 6 or 7. B. Observation of the bare body. C. Trophoblast biopsy of bovine embryos between days 1 and 15. And D. Halved embryos on day 6 and 7. Of these four methods, only trophoblast biopsy on days 12 to 15 and embryo bisection on days 6 or 7 can be considered practicable. The first excludes the possibility of long-term storage, while the second requires additional expense and expertise. Embryo sexing using immunological techniques has also been reported. Experimental evidence suggests that XY antigen is present in early embryos of the bovine, and that serological reagents that identify XY antigen can be used to identify males and females in embryo systems. Now, the sex assessment exercise number 1 is there. You can list any 3 methods of predetermining sex. The sex chromatin. The sex chromatin is a well-developed DNA positive chromocenter observed in the nucleus of most cells from certain tissues in most mammals. It represents a heterochromatic X chromosome which is genetically inactive. It usually lies against the inner surface of the nuclear membrane or adjacent to the nucleolus. The maximum number of sex chromatin bodies in a given interface nucleus is 1 less than the number of X chromosomes. Thus, XY or XO individuals are sex chromatin negative, while XX or XXY have 1 sex chromatin body per nucleus. In farm animals, sexual dimorphism, as indicated by presence of the sex chromatin, is detected mainly in neurons of the brain and spinal cord. The sex chromatin is also detectable in fetal cells from the amniotic fluid of cattle, sheep, and pigs. Conclusion. This unit attempts to give a brief overview of sex determination and manipulation in animals, the sex chromosomes, and which of the sexes are the sex determinants have also been outlined, as well as some processes of sex predetermination. Now, if you have not gotten the answers to self-assessment exercise number 1, which is 1.1, is 3 methods of predetermination of sex, must include flow cytometry procedure, measurement of DNA in separated X and Y bearing sperm population, centrifuging of semen. In summary, sex determination and manipulation in farm animals is a complex technique. Some require specialized equipment, while in some species the males are heterozygous, in others the females are heterozygous. How is sex determined? That is your tutor-marked assignment. How is sex determined in fishes? Now you have a set of references that you can refer to for further studies.